Yes, Omodara, you are most welcome. The floor is yours to kick off the presentation. Thank you.
All right. Hello, everyone. This is group presentation from Group 20, having members of myself, Omodara Ayomide, Toniloba John Adekale, Adedoye Obadare, and Erick Kibirige. Our project topic being combating cassava root rot disease in Nigeria through early detection in Fusarium species using engineered E. coli. So for a background introduction into the topic, taking the geographical area into study Africa, Nigeria to be precise, located in the West African part of the continent. For a background, using cassava as our specific subject matter. Botanical name of cassava being Manius esculentum, a staple crop in tropical and subtropical areas, primarily known for its tattive tuberous roots. Diseases associated with the crop are root rot disease, cassava mosaic disease, bacterial blight, and anthracnose, affecting cassava production. The main symptoms of we've taken root rot disease as the major disease for cassava. The root rot is most poorly understood and studied. It occurs on the ground where they are not visible as much as such their impact is not detected as early as other foliar pathogens. The main symptom of the root disease is a breakdown in tissue of mature tuberous roots with notable change in color and is a foul odor. Tuberous roots are primarily infected by the Fusarium species, and they are the fungal complex that causes cassava root rot everywhere that cassava is cultivated, thereby reducing yield and impacting food security. While resistant cultivars are most effective methods of managing root rot, the genetic basis for resistance remains poorly understood. This research is aimed at providing a more general and adaptive solution to various cultivars, adopting methods rooted in synthetic biology. The general objective is to combat cassava root rot disease through early detection of Fusarium species using engineered E. coli. Now, specific objectives to develop engineered E. coli strain to detect Fusarium species, validate E. coli detection system, and carry out field testing and optimization. Why adaptive solution can be provided? Detection of root rot early is crucial to prevent widespread losses. The current methods available often rely on visible symptoms which may occur late in disease progression. Understanding the molecular response during infection can reveal candidate genes associated with resistance. In this research, synthetic biology techniques are employed. E. coli bacteria is engineered to act as biosensors for Fusarium's presence in the root tuber. These modified E. coli cells detect specific molecules released by the fungus when it reacts with cassava roots.
All right, thank you very much. So let's look at the engineering process. I'm Erick Kibirige. So in our engineering process, we're going to look at identifying, first of all, we shall identify the target species, that will be the Fusarium species. Then we shall use a detection mechanism where we shall develop a genetic circuit. So the Fusarium species, as we had in our background, is one of the major pathogens that attacks the cassava plants. So we shall engineer E. coli to be used for detection. And why we're using E. coli? Because of its wide genome and its variety of proteins. So in our genetic circuit, we have what we call the promoters. So the promoters, we shall use the metabolite responsive promoters. Then we shall have also the transcriptional activator, which will be introduced to specifically interact with the metabolite responsive promoter. And then that will help us to specifically to select and have specificity for the metabolites of interest. Then our reporters, we shall have the the reporters will act as a detection system. And then in our signal amplification, we shall use the digital droplet PCR, which will amplify DNA sequences that are associated with the presence of the Fusarium metabolites. And then now when you look at the validation, we shall have laboratory validation, which we shall verify the specificity and sensitivity to Fusarium species, the biomarkers. Then we shall also do what we call field testing, basically to confirm the effectiveness in the real world setting. And our study area will be Nigeria. Then we shall also develop a detection platform. So we shall have a prototype. Then finally, we shall do monitoring and feedback that will involve to help us in continuous improvement. And also we shall gather feedback from the farmers on how our prototype is being used, and that will help us to improve its accuracy and usability. Thank you. The next one can present.
My name is Obadare Ayomide, and we're still talking about combating cassava root disease with engineered early detection. So starting with advantages of the solution, the first one there is improved crop yield. So by detecting the disease early, farmers can take prompt action to prevent its spread, reducing crop losses and increasing yield. This leads to improved food security and income for the farmers. As in the second one is reduce chemical use. So engineered E. coli can detect the disease at an early stage, allowing for targeted treatment, and then this reduces the need for chemical fungicides, which can harm the environment and then the human health. So they reduce chemical use. There's also enhanced decision making. Real-time monitoring enables farmers to make informed decisions about crop management, reducing their guesswork, and improving resource allocation, which leads to more efficient and effective farming practices. The next one is cost savings. Cost savings, due to early detection and treatment, we can reduce the economic burden of cassava root rot disease on farmers and the agriculture industry. This includes reduced costs for chemical treatment because they are stopping the use of it, then reduce cost for labor and reduce cost for crop replacement. Next one, environmental benefits. Reduce chemical use and improved crop management can lead to more sustainable and maybe eco-friendly agricultural practices, reducing the environmental impact of farming. So there's no washing of chemical fungicides into the streams, killing the aquatic life there. So that's what the advantage of it is. And then improved food safety, reduce fungal toxins in cassava crops can improve food safety for consumers, reducing the risk of the toxins or mycotoxins related to human issues. Disadvantages, okay, high development cost. Engineering for detection requires significant investments in research and development, including lab equipment, personnel, and testing. All this can be very, very costly and expensive to maintain. Next one, regulatory orders. Strict regulations may govern the use of genetically modified organisms, that's GMOs, in agriculture. So there's still regulations concerning that, and it can delay implementation and increasing costs. Next one is public acceptance. Some farmers and consumers may be hesitant to adopt the genetically modified based solutions. So yeah, that concludes it. Thank you.
Okay, and for the final part and are you hearing me? I'm talking about computing technology and the biosafety rules. So for the computing technology, I've highlighted two computing technologies which have been previous ways of setting for cassava rows, and then why the method we have proposed is better. We have the visual inspection. Visual inspection involves going to the farm and manually checking each cassava plant for errors. probably stunted growth, yellowish leaves, foul smell, and some other things. For that, it requires manual efforts, and then it's usually time intensive. And then to reduce that, we can use the method proposed. This will show us a visual sign. For the second one is DNA-based testing. This is not prone to human error and it will work perfectly. The only issue we have there is that it is expensive, and then most farmers do not have the funds to fund things like this. And then for the biosafety, next slide, please. Yeah, for the biosafety, I've also highlighted 2 points. Accidental release could happen, and then if accidental release happens and then the microorganism being used is part of the environment, it will also affect the ecosystem. So my solution to this for accidental release could be to use weakened strains of the E. coli, which cannot function outside the isolated environment. Let's say the isolated environment is the box. And maybe the E. coli gets outside that box, it becomes useless, and then it will not affect the ecosystem. For the second one, the antibiotic resistance. This involves the removal of antibiotic resistant genes from the engineered bacteria in order to prevent the spread of antibiotic resistance. And then we also establish strict protocols in order to maintain strong ethics. How do we do it? How do we handle it? What are the rules involved? What are the punishments involved if we don't follow this rule? In order to preserve our ecosystem. And then the next For the next slide, we have our references where we brought information from, all the links we use in getting the information necessary, and what we use for this slide. And then I believe at the end we have our name. Thank you very much for listening, and I believe I've been able to show you why using synthetic biology to combat cassava. Thank you.
Thank you so much, everyone, for that wonderful presentation. Yeah, and for your time and for the extensive reading that you have done. Thank you.